welcome everybody to today's edition of the webinar on emerging topics in biomolecular magnetic resonance. We have two fantastic speakers today, and both actually agreed to recording. So there's no need for you to record because we will upload the lectures to the ICMLBS YouTube channel. Just to remind everybody, don't use the chat box, but do use the Q&A box for putting your questions. And I mean, you can already put your questions during the talk. That's a nice thing. We can also sort of allow you to speak if you raise your hand. And please stay on for the unofficial part to really discuss all the questions which haven't been discussed before. And I remind you of the early career researcher edition of the webinar, which is ongoing. So without further ado, then I hand over the word to Gerhard Wagner, which agreed to introduce our first speaker of today.
Good morning, everybody, and good evening, Gottfried, or good early night. It's a great pleasure to introduce Gottfried Otting, with whom I overlapped for a couple of years when I was still in Zurich. So Gottfried got his education and studied chemistry at the University of Freiburg in Germany, and then it was PhD at the ETH with Kurt Wittrich, and at that time I overlapped with him. I think he came there in 1984 and I left 1987. We have still a couple of papers together where we worked on T1 noise and things like this. But then Gottfried did very well working there for his PhD. He completed his PhD, I think, in 1987. And then he got a low-level faculty position, a Privatdozent at the ETH, where he focused on water interactions with protein. He established his field, was quite successful, got fantastic papers on this. He worked with Dr. Lipinš from Latvia, and he got elected in the Latvian Academy of Science. Congratulations to this. And this, he worked a lot on this water interaction, protein hydration. And he also worked on protein-DNA interactions, worked on the first homeodomain complex with DNA together with Walter Gehring and Kurt Wittrich. And then he got a position at the Karolinska Institute in Stockholm in 1982, where he stayed for 10 years. And then due to his family connections, for family desire, he moved to Australia, a very beautiful place. And if you have a chance to visit him, you'll be excited what you can see there, and get tips how to find nice beaches in Australia. And you can see some marred landscape from the wildfires, and you'll see more about this. And he in Australia, it's an Australian National University, where he is since 2002. He worked on very interesting things like selfie protein expression, adding lanthanide tags to proteins, and uses for structure determination of complexes, but also for drug binding to proteins and drug design. And he will tell you more about this. And Gottfried, please go on and give your lecture. Thanks very much for staying up so late. I understand it's midnight in Australia. Please go on. Thanks. Gottfried.
Thank you, Gerhard, for the kind introduction. I hope you can see my screen. The ANU is the Australian National Universities. It is in Canberra, and 1.5 years ago, just about for where I live, the view looked like this. And what it is a bushfire that raged for 1 month. It couldn't be extinguished until it rained again. This was one of a very violent bushfire season that burned an area in Australia as large as half of Germany. The fires in Australia are not unusual, and in fact, eucalyptus trees come back after a fire, and some seeds actually require fire in order to sprout. However, this fire was hotter than usual, and it burned large areas of landscape to a degree that trees will not come back for many years. So what I conclude from this is that global warming is real, that we ought to do something about it, that CO2 is a problem, and that we should as a community, maybe avoid unnecessary conference travel in the future. Now everyone is itching after COVID to go back onto a plane, but I think there are strong arguments against this. So I'm really grateful that you have organized this series. I think this is something that I do hope will stay, even though it is midnight in Australia right now. So I would like to show you a few examples of how I think we can use unnatural amino acids or non-canonical amino acids as site-specific probes for NMR spectroscopy. And it was Peter Schulz who introduced about 20 years ago a way how one can genetically encode amino acids. So you can, by the right DNA design, determine where your unnatural amino acid, your non-canonical amino acid is supposed to end up in the protein. And the way this works is that you look at an AMBER stop codon that usually does not code for an amino acid, find the suppressor tRNA, find an enzyme that charges that suppressor tRNA with the amino acid that you are after, and off you go. And the one that we have worked with a lot now is a derivative of lysine, which looks like lysine but is extended and has a trimethylsilyl group at the end, or a TMS group. So this can now be encoded into proteins. That is a system that was developed by a collaborator of mine, Thomas Huber, and his PhD student, Hao Chen. And what it gives then is a signal at 0 ppm, like TMS groups are. It is very intense because it is 9 protons and they do not couple to other protons, so you end up with a singlet in a region of a spectrum where proteins hopefully don't have many signals. So what does it look like? Here's an example, a 19 kilodalton protein, the wild type protein. It looks like a one-dimensional protein in mass spectrum, but with a TMS group, it's clearly there. If you go to a bigger protein like DNA-B, which is 320 kilodalton hexamer, the signal is still quite clearly visible at 0 ppm. So we don't need to deuterate and we can see that signal. Now, what is it actually good for? I mean, ultimately, we want to use it as a site-specific probe, for example, to find something out about ligand binding. Now, if you have a long and flexible side chain, that's great. It means that the rotation correlation time of a TMS group is faster than that of a protein. But if it always sticks out into the water, the environment will always be water. It should always be the same. However, when we incorporate this amino acid into two different sites of the same protein, and this is now a 71 kilodalton dimer, we see different sets of NOEs. So obviously, these NOEs are not just from the TMS group down to the side chain of the lysine, but somehow it makes contact with the protein. And you can visualize this by the fact that the TMS group is hydrophobic. So if you have a hydrophobic patch on the protein, you would imagine that sometime there is a contact between two hydrophobic partners. So we have the best of two worlds. We have a narrow signal because the TMS group is at the end of a long and flexible side chain. It's very flexible. It has a short correlation time, yet it interacts with the protein. Now, here is a protein, there's a crystal structure. It's actually an amino acid tRNA synthetase, so one of the enzymes that charges suppressor tRNAs. And the ligand that we have is cyanophenylalanine. So each monomer in this dimer has one binding pocket for cyanophenylalanine. And we put the TMS lysine into different sites. So 117, 160, 28. When we look at the 1D NMR spectrum, the wild type protein, of course, we don't see the signal at 0 ppm, but then the moment that we have say 28, the position 28 gives us a very nice sharp singlet. It's very exposed on the protein. 160 gives us a singlet. 117 oddly gives us two signals. And that doesn't really make much sense because we always sort of imagine the crystal structure is symmetrical. The two TMS lysine groups are too far apart to contact each other. So why is it actually two signals? So what that must mean, in my view, is that we have that the two pods actually don't fit quite together. There are two different conformations there at the interface that are sensed by the TMS lysine that is closest to the interface of the dimer. Now, the moment that we add cyanophenylalanine, the situation changes. What was two signals originally at the ratio of 1 to 1 has become one signal and doesn't change any further. So then we actually do end up with a symmetric dimer. We can use these titrations to determine binding affinities, of course. So here, this is what we see at site 160. It's in slow exchange. The binding is quite tight, and you see the original signal disappear as we add ligand, and the U1 appears. You can plot the appearance of the signal as a function of ligand concentration. You get a binding curve. It doesn't quite fit. It fits much better if you use a hill plot that has an additional parameter, admittedly, but it does fit much better. It suggests that there is positive cooperativity in binding between the two monomers in the dimer. So this is all nice, and it can be done with fairly low protein concentrations. But could we also use this TMS group to get structural information? And to do this, what we have done is we have made a cysteine mutant, and we mixed it with a TMS lysine mutant. The cysteine we derivatized with either nitroxide tag or the gadolinium tag, and that's paramagnetic. And then we hope that the signal of the TMS group disappears. What disappears is half of the signal. That makes sense because we have a 50% chance of having a dimer where one is paramagnetic and the other one is diamagnetic and has the TMS group. But it disappears only with gadolinium. It doesn't disappear with a nitroxide label. And that can be explained if you look at the equations for the PRE, for permanent relaxation enhancement, and that is simply proportional to S for electronic spin, and for nitroxide that's 1/2, for gadolinium that would be 7/2. So the term is 21 times bigger for a gadolinium than it is for a nitroxide. We have a correlation time, and that is now between the paramagnetic center and the TMS group, and that we don't know. It's going to be faster, shorter than for the protein, because this is not the rotation correlation time, this is the correlation time of the intergroup vector, if you like. And of course, the TMS group is very flexible, and so are the tags that we attach to the protein. And then we have the strongest distance, the strongest dependence, and that's the distance, of course, between the paramagnetic center and the nuclear spin. Now, we can make assumptions. We can say we lose the signal when it's as broad as 300 Hertz. And we can say the correlation time that matters for between two flexible side chains, let it be something, 3 nanoseconds, just a guess. The distance ultimately has really the greatest weight. So then in the case of nitroxide, where S is 1/2, we expect that the two groups have to come within 9 angstroms to actually get this enormous line broadening of 300 Hertz. For gadolinium, not quite as far, because it's 7/2, it's been 7/2, so we can expect that maybe the gadolinium only has to get within 15 angstroms to wipe out the TMS signal. And when we look at the protein, we can of course model what these things look like. And with the nitroxide, we get the red distribution, and with the gadolinium, it's actually very similar with this gadolinium tag. It's a bit broader with gadolinium because the tether to the cysteine is longer. Now, for TMS, for the nitroxide, 9 angstroms is actually not really populated. So we are not surprised that we can still see the TMS signal in the presence of the nitroxide. With gadolinium, 15 angstroms, okay, a small fraction of the populations of possible distances is within 15 angstroms. If you multiply this by 1/r to the 6th in order to find out what the weighting is, because short distances, of course, have much larger effects on the PRE, then we get the blue curve. And then 15 angstroms really suggests that most of the effect is really captured by those conformations that are within 15 angstroms. Now, this is nice. It's not really a proper distance. What we would like is a distance between background atoms. In this case, it happens to be that the background atoms are 17 angstroms apart, but we can't tell this from these PREs. It would be better if we had a direct contact, something like an NOE. Now, can we measure that? And yes, we can. And here's an example. So we can put 2 unnatural amino acids into the same protein chain, one in response to an amber stop codon, the other one in response to an opal stop codon. The tertiary butyl group appears at 1.3 ppm. That's a bit inconvenient because there are protein signals, but we can observe them because it's 9 equivalent protons. It's a singlet. It's very intense. Now, this is a protein that binds arginine, and it has 2 conformations. Without arginine, it is, according to crystallography, open. With arginine, it is closed. Now, with arginine, we expect the way we incorporated these 2 unnatural amino acids that the distance is short, and we might see an NOE between the tertiary butyl group and the TMS group. In the absence of arginine, we expect that the distance is long and that we don't see the NOE. Now, at the top, you see the spectrum without these unnatural amino acids, at the bottom with, and also with arginine. So this is a close conformation, and we see a peak below 0 ppm, that's the TMS, and one at 1.3 ppm, and that's the tertiary butyl group, and hopefully there is an NOE. But in the absence of arginine, in the absence of ligand, where we expect the open conformation, we still see the closed conformation. So this protein actually, without the ligand, is both open and closed. Half the time closed, half the time open. But also, the tertiary butyl group, when you look carefully, has 2 peaks. So we should run a nosy experiment, and so we did. Here on the right, you see, in fact, with arginine, there is an NOE between the tertiary butyl group and the TMS group. And this is in a 32 kilodalton protein without deuteration. It's nice to observe. Without arginine, we get 2 peaks on the diagonal, and when you look closely, you see the exchange peaks. So we can measure the exchange rate between open and closed conformation, and the closed conformation indeed also makes the NOE as it did in the presence of arginine. So these nice narrow signals are useful for measuring NOEs. In conclusion, I think TMSK is a great tag for large proteins. It's easily observable. It gives you maximum sensitivity. You can use a cryoprobe that you already have in the instrument. It's great for measuring small KD values because you can use it at very low concentration. We have measured KD values in the high nanomolar range in very slow exchange. Now, today it's very popular to use fluorine NMR as tags. And if I compare the 320 kilodalton DNAB hexamer with the TMSK, that is the signal here at 0 ppm, it is, what's the width, 0.05 ppm maybe. I can incorporate trifluoromethylphenylalanine and get a line width of half a ppm in the fluorine NMR. I think I prefer the proton. But anyway, fluorine is very modern. So since Hari Afanari and Gerald Wagner published very nice work with an aromatic fluorinated carbon-13 labeled compound, we looked at indole. So Ansis Belekis works in Latvia at the Institute of Organic Synthesis. He synthesized this particular indole. And Iresha Herat, who works in my lab, discovered that using self-reporting synthesis, the E. coli cell extract still has the enzyme that's necessary to convert an indole into tryptophan. And she can incorporate this into T4 lysozyme that has 3 tryptophans. You can then measure the carbon-13 1D NMR spectrum, and as Hari Afanari has shown, you get beautiful Trosi effects. So you have for 3 tryptophans 3 narrow lines and 3 broad ones, and these are just separated by the 1-1 coupling between the fluorine and carbon. But Ansis, on the way to the fluorinated indole, also made the indole, just labeled site specifically with carbon-13. And there the line widths are greater, and there are 3 narrow lines, 3 very broad lines in the carbon-13 1D NMR spectrum. Now, we wanted to apply this to something, and the system we applied that to is a metallobeta-lactamase IMP1. There are various crystal structures, and here in blue and green are 2 crystal structures with and without an inhibitor, captopril. And then in pink is another structure without the inhibitor, and it is a bit different. So we wanted to know, is this an active site loop, a flap that closes when the inhibitor comes, or what happens actually in solution? And to do this, we incorporated cysteines at different sites of the protein and attached paramagnetic tags to generate pseudo-contact shifts for the tryptophan. Now, I won't tell the details about pseudo-contact shifts, that has been reviewed many times, but this is a tag that we attached at 3 different sites. They generate pseudo-contact shifts that are positive for the blue areas and negative for the red ones. And we want to measure these pseudo-contact shifts at the site of the indole to find out where exactly it sits relative to those tags, which we know where they sit relative to the protein. Now, we've measured the pseudo-contact shifts with and without inhibitor for the 3 different tagging sites. The indole that we're interested in is number 28. It is carbon-13 labeled at a single site, And this gives us sufficient resolution to measure all these pseudocontact shifts. So in red, it's with terbium, which shifts to one side. With thulium, it's blue, that shifts the signal to the other side. And in black, it's with a diamagnetic reference. So we can combine all this information to find out and try to figure out where the indole is in the protein. And it turns out that for the CH, it's all these red and magenta balls, and the crystal structure is within the range that the NMR data permits. And the NH we can also observe, and also that is in the range that the NMR data permit. So then we looked also at the data with the captopril inhibitor, and we found that again the crystal structure was perfectly fine, and NMR data are compatible with it. So it seems that in this particular case, there isn't much movement of the flap when the inhibitor comes and binds. In summary, carbon-13 proton-labeled indole facilitates the selective observation of tryptophan side chains. We had to deal only with a single signal per tryptophan residue. And the E. coli cell-free extracts can make fluorotryptophan from fluoroindole, which is convenient. Here's another unnatural amino acid. Actually, it is a natural one. It is phosphoserine, and it can be incorporated again in response to an amber stop codon at any site that you wish. In this particular case, we used GB1 and we incorporated 2 of these residues. Now, between the 2 phosphate groups, you generate a lanthanide binding site, and in red we have added terbium, in blue we have added thulium. And we know, of course, the structure of the protein. We can fit the expectations, so we can calculate the PCS and compare with the experimental one. And we get a correlation that pulls the socks off when I look at it first. So the quality factor is less than 5%. It is a stunning correlation between experimental and predicted data. Now, we published this in a journal, a new journal that's called Magnetic Resonance, an open access journal with affordable fees, which I would like to advertise here with. The last bit that I would like to talk about is PREs. Many people use PREs simply by attaching a paramagnetic tag and then looking at line broadening or peaks disappearing. And the distances that one gets out of this, in principle, are very nice. In practice, often they're quite polluted by intermolecular relaxation enhancements. So how do you know that the PRE that you have generated is just intramolecular if you have lots of paramagnetic molecules swimming in solution? The reference that we use is a diamagnetic molecule without the paramagnetic tag. And the solution that we'd like to propose is to use an internal diamagnetic reference, that is, have a diamagnetic compound and a paramagnetic compound simultaneously in solution at the same time, and measure the relaxation rate for each of the components. Now, you can do this only if you have separate chemical shifts. So therefore, you need a lanthanide ion that also shifts the peaks that generate pseudocontact shifts. You can do this with erbium, you can do it with terbium. Erbium is better because it actually doesn't generate large pseudocontact shifts. If you generate large pseudocontact shifts, you will also get RDCs, and they broaden the lines in the paramagnetic state. That's not very nice. But if you use erbium, you don't get that many. If you use a low field, then RDCs are also small. If you use short relaxation delays to measure your PRE, you also end up with more accurate data than if you wait for the RDCs to evolve. So this is the recommendation: use an internal diamagnetic reference. Then, in order to resolve the peaks, use the 3D HNCO, say, and then we were able to obtain with calbindin D9K, which binds the lanthanide ion at a very defined position, actual distances that fit over a range of something like 12 to 24 angstroms. And I think this is something that is very hard to achieve any other way. So with this, I would like to thank all the people who have done the work for this. Thomas Huber, who established a very efficient selection system together with his student Hao Cheng for amino acid DNA synthetases for a number of unnatural amino acids. Mithun Mahavatha, who worked with the incorporation of phosphoserine Henry, who is the expert in all things paramagnetic in the lab, Iresha, who found a way to incorporate indol that has been synthesized with great pain by Ansis Melekis, using only very small quantities of it because you can use cell-free protein synthesis to incorporate it. Then Bim Graham for making tags, Luke Adams who synthesized the TMS lysine. And I thank you for your attention.
Thank you, Gottfried. So you certainly will answer questions. I just read some questions from the Q&A. Elon Slens asks, is it necessary to add any specific tRNAs and transamino acyl transferases during the protein production in order to incorporate unnatural amino acids? Of course you need, yeah.
Yes, of course. And the suppressor tRNAs are generic. So once you have a tRNA that recognizes an amber stop codon, you only have to worry about the amino acid tRNA synthetase that attaches the amino acid at the other end. And that synthetase has to be specific. And the way you find that a suitable amino synthetase is that you have to make a library of mutants as large as possible, and then a selection system to find the few mutants or the one mutant ultimately that actually recognizes your unnatural amino acid specifically, doesn't recognize a natural amino acid, and is capable of incorporating that, of attaching this unnatural amino acid efficiently to your suppressor tRNA. But this is a system that has been worked out by Mehl, for example, by Peter Schulz, and also in other laboratories.
May I have follow-up questions? Do you get unwanted termination of synthesis if you use a stop codon instead of getting the unnatural amino acid?
Well, nowadays there are cell lines, E. coli cell lines, where all amber stop codons have been eliminated and replaced by other codons, other stop codons. So then the amber stop codon is always recognized as a valid codon because also the RF1 termination factor has been eliminated from those cell lines. And we use that regularly, a strain developed in Japan, B95, that gives good incorporation rates.
Thank you. David Eliezer asks, can you comment on introducing similar labels, multiple highly mobile protons, by modifying cysteine residues to avoid the complications of unnatural amino acids? Yes,
and of course you can always derivatize cysteine residues, and that's done frequently to attach, for example, fluorine tags, or we have done it also with the TMS tag. The problem is that then your protein must not contain more than a single cysteine residue. If you have cysteines that are essential for catalytic activity, for structure, you need selectivity. And one way to get this selectivity is maybe if you can incorporate a selenocysteine at a single site that is much more reactive than cysteine. Adachi, that you can see in the background, she has worked with this. But the yields are not great, so it's not a very smooth system yet.
There's an anonymous attendee. The diamagnetic reference is a protein tagged with quenched spin labels, not untagged protein.
This is a very interesting question. We always try to make a diamagnetic reference with a compound that is the same as a paramagnetic one, but diamagnetic. So with lanthanides, you can use yttrium as a metal instead, or lutetium or lanthanum, and that would be diamagnetic, the same environment. For nitroxides, one would reduce maybe with vitamin C and get hydroxylamine instead. In practice, it often turns out that the spectrum with a diamagnetic tag is very, very, very similar to the one without tag. So we still attach the diamagnetic tag, but sometimes the students sort of think, oh, do I really have to do this? So the diamagnetic reference is important in principle, in practice not always required. It depends on the detail of information that you want to extract.
Till Friesewinkel asks, are unnatural amino acids an option for introducing lanthanide binding sites in proteins?
So there have been some metal binding amino acids published. They have only very few coordination sites for proteins, for metal ions, and they invariably tend to precipitate the metal ion, the protein. So what you can do is you can put amino acid into a protein, say, that has an azide group. Then you can use a tag that has an alkyne group and use click chemistry to attach the two. But that needs a copper catalyst. Half of the proteins that we have played with in this way precipitate the moment they see copper. So it is not straightforward. The phosphoserine is not, you can treat it as an unnatural amino acid and it generates binding sites. But also there you have few coordination sites. So you end up with the possibility to precipitate proteins the moment you add lanthanides.
Another question, anonymous. If I can see PCS, could it be because of too flexible lanthanide binding tags?
That's an excellent question. Yes, if the metal ion reorientates, or the complex that contains the metal ion reorientates with relative to the protein, you very rapidly average the PCSs to zero. The PREs remain, relaxation enhancement remains, but the PCSs disappear. So then a whole cottage industry has developed where people try to develop tags that have a short tether that keep the lanthanide in a defined orientation relative to the protein. And for example, Daniel Heusinger has a very nice tag which generates large superconic shifts. It has a very rigid tether and it works. But the synthesis of it is not for the faint-hearted.
I have a question. So sometimes when we try to get fluorinated tyrosines in proteins, sometimes the proteins don't fold. But tyrosines are more internal than tryptophans, or do you see this problem? For fluorinated tryptophans would be very interesting, but maybe a problem.
Yeah, so it's true. All the proteins that we have tried with fluorinated amino acids more prone to precipitation. And tryptophan, I think, has been around for a long time. Fluorotryptophan doesn't really precipitate maybe as easily as other aromatics, I believe. Yes, absolutely. The fluorine is not the same as hydrogen, and you have a greater chance of precipitation.
I don't see any more questions, and thank you very much. Very interesting talk, and we'll learn from it. Thanks very much, and thanks for staying up so late.
Thank you for having me.
So Beat, then it would be your turn.
Thank you. Well, it's a pleasure to introduce Professor Isabelle Marcotte from Montreal in Canada. And she got her master in chemistry at the Université de Montréal in Canada before she made the doctorate with Michel Auger at the Université Laval. Then she moved to Switzerland between 2003 and 2006. She did a postdoc in my group, and we were at that time mostly interested in spider silk and the order, the local order and the overall order in real threads of spider silk. After the postdoc, Isabelle went back to Canada. She went to the University of Quebec at Montreal, and she went through the ranks as an adjunct professor, an agrégé, an associé, a titulaire professor, and she became then director of the department in 2016. Since 2017, she also, in addition, is the vice president for research for the Faculty of Sciences of the university. She has been still going on with some research which is related to the silk, namely the mussel silk or the mussel byssus, which is the material the mussels produce to adhere themselves to the ground in the sea. But she has changed also quite a bit the research directions, and she looks now at membranes and intact bacteria and microalgae. And that's what she will report today. The title of her talk is Identification and Architecture of Polysaccharides in Microalgal Cells, an in situ solid state NMR approach. Isabelle, I'm looking forward very much to your presentation.
Thank you very much, Fiat, for the very nice presentation. Maybe it shows that I'm a little bit hyperactive, working on many different systems, but they're all connected in a way. So I'm sharing my screen now. Do you see it? Oops, sorry, I'll go back to the beginning. Okay, y'all see this, the slides, yeah?
Yes, we see them.
Okay, so thanks for the organizers for the invitation to give a talk today. It's so good to see everyone from the NMR community. Yeah, as Beat was saying, over the last few years, we've been very interested in developing approaches to tackle in-cell NMR. And so today I'm going to present what we've been doing on microalgal cells. So as you know, microalgae are unicellular microorganisms. There are about 30,000 species identified so far in fresh and saline water. They're a very important organism because they support the aquatic food chain. And so the effects of human activities on microalgae can impact all the trophic food web. They're also an alternative food source, and because of their nutritional and energetic value, they have a lot of proteins and sugars and lipids that are interesting. And they're also exploited in biotechnology as agriculture feedstock for biodiesel production, cosmetics, nutritional supplements. So cultivation parameters have to be controlled to optimize the biosynthesis of high-value molecules that we can then maybe exploit. So it's important to provide tools to characterize microalgal cell components. So the objective of the work I'm going to show today was to establish in situ solid state NMR methods to identify specifically today's sugar and sugar-bound molecules and to determine their atomic level organization in fully labeled microalgae. So why sugar? Because they have a very importance since they're very important in the structure of these organisms, and they're also a source of energy. And we're going to focus today on Chlamydomonas reinhardti, that I will call Chlammy to make it short, which are those little microalgae here. And this work has been carried out by a very brilliant PhD student who's shown here. His name is Alexandre Poulazan, and I'm going to show the work he published on starch in 2018, and also the ongoing work that we're preparing for submission. So let's start with showing Chlammy. So Chlammy is a freshwater microalga, and it has different carbohydrates in different locations. So in starch grains that are used to store sugars, there is sugar reserve in the cell and organelle membranes where we find galactolipids, and also in the cell wall where there are glycoproteins bound to oligosaccharides. So we will first focus on starch. Starch is a semi-crystalline network of amylose and amylopectin chains that are shown here. They're alpha-1,4 glucans. Starch is a double left-handed helix. They're made of double left-handed helices held by hydrogen bonds. They are different. There is an amorphous form, and there are different crystalline forms, actually 3 forms. The A starch, which has a closed packing, and the B starch, which has an hexagonal packing and which is more hydrated. So as a first step, we wanted to perform a complete and unambiguous carbon-13 assignment of A and B starch. And to do that, we used different starch before going to study the whole cells. So we used type A starch, which comes from starch grains extracted from amylopectin-rich Chlammy mutants. with a 70% crystallinity determined by XRD. Type B starch, which is obtained by starch retrogradation from the wild type CRNRT, which has a 55% crystallinity, and amorphous starch, which comes from freeze-dried amylose-rich starch produced by another clamine mutant. So you can see here the result obtained. These are just 1D spectra of the E-type, B-type, and amorphous starch. And I will just use another pointer. Okay, so you can see here the glucose C1 and C4 and C6 carbons are readily resolved on the spectra. Their numeration is shown here. And C1 and C4 are very important carbons because they allow the identification of starch types, because they're more sensitive to conformational changes than other carbons. There are splittings here of the C1 carbons, and they reveal different crystalline forms. These are due to different spatial arrangements that lead to 3 possible environments for carbon 1 in the A form and 2 in the B form. We also observe a broad distribution of conformations in the amorphous starch, which is shown by the large chemical shift dispersion of the C1 peak. And on these 1D spectra, the C2 and C5 peaks could not be distinguished. So that's why we moved on to 2D experiments. And we chose the 2D carbon-13 inadequate because it provides through bond connectivities, and it improves the chemical shift dispersion through the double quantum dimension. So you have here again the type A, type B, and amorphous starch spectra. And they allow the assignment of the C2 and C3 and C5, and C5 peaks. And also, they allowed to establish a new difference between the A and B type starch via the C5 splitting here, which you can see on the B type that couldn't be seen before. What was also interested was to observe to be able to assign non-reducing terminal glucose group. They clearly show a different spin system. And this is here, these are here normalized traces of the spectra of amylopectin, of the type A starch, type B starch, and amylose. And we can monitor actually the relative proportion of the end groups by closely analyzing these normalized traces. And we can see that there is an increase in the reducing end group content when going from linear, like the pure type B starch, to a branch starch like the pure A type. So these end group here, non-reducing terminal glucose groups, allow us to have an idea of the amylopectin, amylose content, or the branching abundance. So with this part, we were able to assign all the carbons in the A and the B types starch and the amorphous starch, and we were also able to assign the non-reducing end groups, and these chemical shifts are reported in the paper of Alexandre Poulazan. And with those data in hand, then we were able to move on to the assignment of starch in the actual cells. And so this is the inadequate spectrum of of all Chlamydia monocyte cells. And here in green, we compare the starchless strain to the wild type strain, and that allows us to confirm the starch signal. And you can see there is also a very little overlap with other saccharides. And if we compare the chemical shifts obtained from in the wild type in the microalgal cells, if we compare the chemical shifts to what we obtained on the starch before, we were able to determine that the starch in Chlamydia is in an A-type starch. And this is consistent with the high amylopectin content that has been reported. What we thought was also interesting was to be able to do an in-cell monitoring of the variations in the starch content. The starch content is very indicative of stress. And to do that, we used 1D carbon-13 solid-state NMR spectra, and we compared wild type CLAMI and different mutants, one that is rich in amylopectin, one that is rich in amylose, and one that is starch inhibited as a control. We compared a normal growth condition to conditions where there is a nitrogen starvation, shown in blue here. And we can really see that this stress, this N2 starvation, increases the starch production. So altogether, in this part, we were able to show that we could determine the starch type and the overproduction in intact microalgal cell. And this is very unique to solid-state NMR because we don't have to do any starch extraction. This information can be obtained directly in the cells. So now I will move to the other part of my talk, where we will focus on the cell wall of CLAMI. So CLAMI cell wall, as shown by electron microscopy, is a network of ramified fibrils and globular proteins. And it contains hydroxyproline-rich O-glycosylated proteins. And this hydroxyproline binds with chains that comprise mannose, arabinose, galactose, glucose, rhamnose, and xylose sugars. So before doing any experiment on the actual cells, we first worked on cell extract, so to make the assignment easier before going into towards a more complex system. So first, what I'm going to present is obtained from cell wall extracts. So we carried out the assignment of the amino acids using carbon-13 top C and high field inadequate. And the assignment was aided by the biological magnetic resonance data bank. And this is the spectrum with all these assignments, but actually 96% of the peaks were assigned. That is amino acid and sugars included. And the hydroxyproline signals were unexpectedly low compared to what's revealed in the literature, but there is about 1.5% abundance of hydroxyproline in our sample according to mass spectrometry. By comparison of these chemical shifts to chemical shifts values, such as in the chemical shift index software, we were able to have an idea of which conformational environment environment we could find these amino acids. And you can see here all the different amino acids, and in blue, that's random coil, and in red, it's beta sheets. So altogether, we find these amino acids mainly in random coil environments, and we find the presence of beta sheets. There are no alpha helices. So now knowing about the amino acids, we turn to the sugar assignment, and we use again this top C and inadequate experiment to identify all the sugar peaks, and we used for that the complex carbohydrate magnetic resonance database that has been established by Professor Joe Wine. And this is again another spectrum showing all the assignment of this sugar. And we can see that there's a lot of mannose, of course, but we see rhamnose, xylose, arabinose, and so on. And according to our NMR data, but also to mass spectrometry data, we find that mannose is the most abundant sugar in the cell wall, followed by arabinose, and then galactose, and then glucose, and rhamnose, and xylose. So with this amino acid and this sugar assignment done, we wanted to look at amino acid sugar contacts because we were looking for glycosylation. Glycans are attached to the backbone via an amide or a glycosidic bond, so they can be either nitrogen or oxygen linked. And all glycosylated sugars, they usually bind to hydroxyproline, threonine, or serine. And here, when we were looking for these contacts, we realized that the amino acid glycan contacts, they were very mobile, they were found in very mobile and flexible regions. And so they were better seen with DNP, so that's the DNP spectrum. And also, they were seen when the microwave were off on these frozen samples, so which really gives us a clue about the mobility of these especially hydroxyproline contacts. And so we were able to detect with the spectrum amino acid glycan contacts between so threonine, and so in purple here, threonine and mannose, and with also glucose and galactose. And in red here, you can see the contacts between hydroxyproline and mannose, as well as arabinose and galactose and glucose. So altogether, our data show that mannose is abundant, and it's clearly involved in contacts with hydroxyproline and threonine, but not serine, not actually in the cell wall of Chlamydomonas canarti. And this O-glycosylation has important roles. It's important for cell recognition, and it can bring flexibility to biomacromolecules such as cartilage and tendons. So clearly for microalgae, this is an advantage for cell division. We have also detected interglycan contacts between mannose and galactose, but I will not show them today. So we further explored, we wanted to know more about this glycosylation. So we decided to chemically deglycosylate the cell wall extracts. And to do that, we used 3-fluoromethane sulfonic acid because it was shown to bring little protein degradation. And with that, we realized that by removing the sugar, there was a little loss of structure. So for us, it's indicating that there is a complex architecture in which the glycans are more abundant in specific regions of the cell wall. For example, we could think about glycan-rich granules. We lost the hydroxyproline and threonine signals, and we wonder if it's because they become too mobile when glycans are not linked to them. But this is something that we have to explore a little bit more. Then we were interested in the hydration of the cell wall, and we did a water-edited DAR, where you can show here the hydrated polysaccharides, the hydrated hydroxyproline, and the less hydrated amino acids. And this hydration is reported here in this histogram. And you can see that altogether, glycans are more hydrated than amino acids, that the amino acids that are involved in glycosylation, they are also more hydrated than other amino acids. So it's like it looks like there is a gradient of hydration going from sugar to amino acids. So these results suggest that water is involved in structures where glycosylation occurs, and this has been demonstrated in other works in starch. So the work in progress now is to carry out the whole cell study. So whole cells are very complex samples. They are signals from all the sugars. So here, that is what we're focusing on. And so far, the assignment has been done, and this was aided by the use of mutants. So you can see here the spectra overlaid of the wild type Chlamys in black, a mutant that has no cell wall, which is in red, and the extracted cell wall, which is in blue. And this was very reassuring to see that there was some overlap between the extracted cell wall and the wild type Chlamys. So we know that working with the extracted cell wall was a good strategy before going to work with the whole cells. We did DNP also of the whole cells. So the spectrum is similar to the cell wall extract and reveals new amino acid contacts and intense hydroxyproline signals. And this is still under our investigation, and hopefully in a few weeks we have a little bit more to say about that. And we're trying now to building a model to understand all these results. And it seems to be pointing to the direction where there would be in the cell wall of Chlamys hydrated sugar granules that would be surrounded by less hydrated protein regions. So, so that's where we're at now, and we're still doing this work. And just to end my talk, I would like to acknowledge all the people who actually did the work. So Alexandre Poulazin, who seems to be looking for microalgae here, Alexandre Arnold and Paul Warszawski, who were very involved in the project, the group, my group members here, Professor Tu Wang, with whom we've been collaborating on this project, and where Alex has been doing a research visit for a while. So we're very indebted to Professor Wang for all his help in this project. And also to the MagLab facility, Dr. Ivan Hung and Frédéric Monting-Vigier, who were very, very helpful for all the DNP experiments and the high field 800 megahertz NMR experiment, and all these agencies for the funding. And I would be happy to answer your questions.
Thank you very much, Isabelle, for a nice presentation. It's now open for questions. I don't see any yet, so maybe I can ask one. You showed these secondary structure plots with beta sheets and random coils. And is the random coil, is this just a coil arrangement, or is it actually related to dynamics?
So I'm going to find the slide. That's here.
This
Okay.
one, yeah.
Yeah, so sorry, can you repeat the question?
Is the shifts that are random coil, are they in parts of the sample where there's more dynamics? Is it really averaging out chemical shifts, or is it just in a coil conformation which is static?
It's a good question, but actually it's true that dynamics is really an issue with these microalgae, because they're heterogeneous, and it seems that that what is best filtered out is starch, for example, and these very rigid amino acids. But altogether, it's a pretty dynamic system. But yeah, because we've been trying different experiments with different looking at different dynamics, we're pretty convinced that it comes from random coil, not from averaging from the dynamics. But maybe that's something we can double check just in case. But that's a good suggestion.
There is a question from Erika Brendler, and she wants to know whether the what is the C13 isotope contents of the samples?
What is the abundance?
Yes, the isotope abundance.
Yeah, it's about, I would say something like 95% at least to 100%. They're very easy to label. We just have to grow the microalgae in carbon-13 label sodium bicarbonate. So yeah, it's almost 100%.
If may I add a follow-up, because I had the same question. because the inadequate experiment in natural abundance probably wouldn't show up. I mean, you need carbon-13 label for that.
Yeah, we need carbon-13 label. Yeah, sure. Otherwise, we would miss so much signals. Yeah, there would be an issue in terms of the intensity of some of the components.
Well, you wouldn't get carbon-carbon couplings essentially because of the natural abundance, right?
Yeah, sure.
There was another question by an anonymous attendee, and he wants to know whether DNP affects mobility of the biomolecules considering cryogenic temperatures of DNP.
Actually, because of the temperature of DNP, it was an asset for us because we think, for example, like hydroxyproline, they seem to be in very mobile regions. So the fact of using so low temperatures, it enabled us to actually see the signals that were otherwise not seen due to the dynamics.
And then there is a question by Rosalie Thompson. Did you see any changes in the C6 carbon of starch? In cellulose, the C6 shift seems to be quite sensitive to conformation, or is it too mobile?
The C6 actually, it's actually not the best one to look at. So it's really like the C1 and the C4. So yeah, C6, there's not a lot of change. So it's around 60.8, 61.5. So I don't know if I answered the question properly. If it was the question, if my answer is related to the question properly, but so actually the best carbons to have an idea of the type is really like the C1, the C4, and yeah, we established now the splitting of the C5.
Marcus wants to ask something, I think.
Please. So could you just comment a little bit on the sample itself? I mean, you pack the microalgae in the rotor and then they stay alive, or and if so, how long? And how do you do that?
So yeah, we did check that. So they survived the experiment. Obviously not the DNP, but if we do, like usually we do the experiments at room temperature, so that means that the sample are about 35 degrees, and they last, and we don't see any degradation peaks. That's something we monitor too, and to make sure that they would stay alive. And we also in the past took some. I didn't show the pictures here, but we took microscopy images, and we really see that the morphology of the cells is intact too. So they doesn't seem to be a lot affected by the magic angle spinning.
And if I may have a follow-up, how much enhancement do you see by DNP for this system?
How much enhancement? I think it's time. I don't know, times 6 or something like that, almost, if I remember well. But we could not really see well the hydroxyprolines before. So yeah, I would say 6 times, 10 times better.
Okay, thank you.
There's another question in the chat which asks from Himanshu Singh. 3 questions. Which starch form is dominant in algal cells? Maybe I take one after the other.
So in the algal cell, it's the A-type. So that's what we discovered, and we were in Clamy actually, yeah, and we were happy to be able to see it in the intact cells without having to extract chemically the starch and then perhaps affect the structure of the starch.
Did you see a correlation between starch and lipid levels under N2 starvation?
Yeah, it tends to build up lipid reserves. So it increases the content in the triacylglycerides. So there are a little bit more of lipid rockets. But we were not looking at the lipids there, so it didn't show the results.
And there's a third question saying, did you record carbon-13 starch spectra on natural abundance or used C13 acetate CO2 carbon precursors?
Actually, these were the starch was extracted from mutants, and we could grow we could label the mutants, so the starch was labeled.
Okay, there's another question. Is there a possibility of amorphous and crystalline type A or B existing in the same cell wall? And how could this be distinguished by NMR due to signal overlap?
The type A and B, that's a good question. If we look at the spectra, I guess if there's an overlap, that's a good question. I guess that, yeah, there would be some overlap, but possibly some of the peaks might be good reporters, like the C5, for example, that has this this little splitting here. So yeah, I think we should be able to see the difference with good resolution.
And then your diver student, Alexandre Boulazin, he makes a remark and wants to help to answer the enhancement question. He says it's 40 to 50 times about if you use Amupol, and it's very easy to hydrate the samples.
Thanks, Alex.
Hi. So I studied the plant cell wall over in Davis in just some biofuel relevant crops. And one way we distinguish between different subtypes of amorphous cellulose is specifically using carbons like 4 and 5, just because they tend to have more distinguishable carbon shifts. But I'm wondering if you've seen any distinguishing peaks for, say, carbon 1 for the amorphous allomorphs of your polysaccharides.
Oh, sorry, I'm not sure I got your question.
Oh, I was just curious. If you've examined the well-resolved carbons that are illuminated in the double quantum experiment to better sort of ascertain changes between crystalline and amorphous subspecies of these polymers.
You want to know if we tried other types of species?
So if you see the crystal and the amorphous phases in the double quantum experiment.
Yeah, it's just a point of it's one way that some cellulose structures can be distinguished using like when things are particularly overlapped. So it was just one point of curiosity.
I think there's always a little bit of amorphous in it, because when we did the XRD, there was like about, let's say for the type A, 75% of crystallinity, for example. So there's some, it's crystalline, but there's always a little bit of amorphous content. I don't know if I answered your question.
No, I was just wondering if you could track a particular carbon within the actual sugar ring in your double quantum experiment, because you do get to see peaks no longer overlapped, given the double quantum coherence just kind of spreading everything out. Yeah.
Yeah, it makes it easier to track the C1, for example. Yeah, for sure.
Okay. Okay. Cool. Thanks.
Okay, I think we see no further questions.
Yeah, thank you very much, for handling it, and thank you to both speakers for the fantastic presentations.